Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. First of all, Assalamu alaikum to all of you. I am Dr. Hadis Shagird. Let's start our topic without consuming our precious time and useless chat. So, our today's topic is transcription. There are three important questions about the transcription. These questions are: what is transcription? Why transcription occurs? And how transcription occurs? If I answer the first question, that what is transcription, the definition of the transcription is that the formation of messenger RNA from DNA is called transcription. It is the simplest definition of the transcription. The modified definition of the transcription is that the transfer of information from DNA to the ribosomes in the form of messenger RNA is called transcription. It is the modified definition of the transcription. If I answer the second question, that why transcription occur, then we all know that our body needs some proteins to perform its functions properly, and we know that these proteins are synthesized in the ribosomes. And we also know that ribosome cannot synthesize any type of protein without any information, instruction, or coding. And this information is present in the DNA. And we also know that the DNA is present in the nucleus. Then how this information will transfer from DNA to ribosomes, so that DNA synthesize its copy in the form of messenger RNA and then transfer it to the ribosomes for the synthesis of the protein. That's why transcription occur. The third question is that how transcription occur. In the answer of this question, we will discuss about the process of transcription. But before discussing about the process of transcription, we have to know about some structures which I am going to discuss with you. This is double helical strand of the DNA. From this point to this point, it is a gene. Okay, the starting point of a gene is called promoter. The site or region from which a gene start is called promoter, and where a gene is end is called terminator. And this middle portion is known as gene. It is an other gene. It is an other gene. If I cut this gene from the DNA and zoom it, it will look like this. It has three portions: 1, 2, and 3. The middle portion is gene. This portion is terminator. And this portion is promoter. Now, what is promoter and what is terminator? Promoter is a site from which transcription will start, and terminator is a site where transcription will end. There are specific regions, specific sequence of nucleotides in the promoter. These sequences I am going to discuss with you. If we consider this point positive 1 and we move left side to right side of this positive 1, it will be positive 2, positive 3, positive 4, and so on. But if we move left side of this positive 1, it will be -1, -2, -3, and so on. Okay, these numbering, I am giving this numbering to the sequence of nucleotides. Okay, suppose after 10 nucleotides, after 10 nucleotides, minus 10 means after 10 nucleotides, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. After 10, after minus 10 nucleotides, a specific sequence of 6 nucleotides is present. This sequence is 1, 2, 3, 4, 5, and 6. This sequence is thymine, adenine, thymine, adenine, adenine, and thymine. This sequence of nucleotide is also known as Tata box. Tata box. It is also known as Pribno box. Why it is called Pribno box? Because it was discovered by a scientist whose name was David Pribno. That's why it is also called Pribno box. After this sequence and after minus 35 nucleotides, another sequence of 6 nucleotide is present after minus 35 nucleotides. And other sequence of 6 nucleotide is present. 1, 2, 3, 4, 5, and 6. This sequence is thiamine, thiamine, guanine, adenine, cytosine, and adenine. This sequence of 6 nucleotide is also known as minus 35 element because it comes after the minus 35 nucleotides. Okay. After the discussing about these structures, I am going to discuss about RNA polymerase, which is a very important factor in the transcription, which is responsible for the transcription. Now I am discussing about RNA polymerase. RNA polymerase basically, if it works in the prokaryotes, only one type of RNA polymerase is present. If transcription occur in prokaryotes, only one type of RNA polymerase is responsible for the transcription. But if transcription occurs in the eukaryotes, there are three types of RNA polymerase which are responsible for the transcription. These three types of RNA polymerase are RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. RNA polymerase 1 is responsible for the synthesis of ribosomal RNA, and RNA polymerase 2 is responsible for the synthesis of messenger RNA, and the RNA polymerase 3 is responsible for the formation of transfer RNA. Okay. Uh, Now I am going to discuss about the structure of RNA. RNA polymerase. Basically, RNA polymerase consists of 6 units. These units are 2 alpha, beta, beta prime, omega, and sigma. When these 6 nucleotides combine with each other, then an RNA polymerase is formed. Okay, RNA polymerase also known as holoenzyme. Like this: 1, 2, 3, 4, 5, and 6. This is alpha, this is alpha, this is beta, this is beta prime, this is omega, and this is sigma. This is a mixture of these small units, 6 small units. Okay, it is RNA polymerase. When this RNA polymerase dissociate, it will convert into two parts. 5 unit, one part is consist of 5 units, and other consist of single unit. These are alpha, alpha, beta, beta prime, and omega. Here will be sigma factor. Okay, it is known as sigma factor. When sigma factor leave the RNA polymerase, the remaining structure is known as core enzyme. This is core enzyme, and it is sigma factor. What is the function of core enzyme and sigma factor? The function of core enzyme: formation of RNA strand and the breakage of hydrogen bond between the nitrogenous bases. Okay, and the function of the sigma factor is recognition of site from which transcription will start. Okay, its function is just recognition of the site from which transcription will start. Okay, if I draw the structure of RNA polymerase like this, This is coenzyme and this is sigma factor. This is coenzyme and this is sigma factor. It is collectively called RNA polymerase or holoenzyme. Okay, when it disassociates into two parts, these parts are like this. This is coenzyme and this is sigma factor. Okay, this is the structure and function of the RNA polymerase. Now we are going to discuss about the process of transcription. There are three steps in the transcription. Okay, the first step is initiation, the second step is elongation, and the third step is termination. Okay, now I am going to discuss about the first step, initiation. In initiation, RNA polymerase comes and attach to the promoter site of the gene. Okay, like this. It is a big molecule. RNA polymerase is a big molecule. It is attached to the promoter site of the gene. This is coenzyme and this is sigma factor. They are collectively called holoenzyme or RNA polymerase. Okay, when it attaches to its to the promoter site, the coenzyme break the hydrogen bond between the nucleotides. Okay, between these heterogeneous bases, like this. When hydrogen bonds between the nitrogenous bases are broken, then replication bubble is formed, like this. Like this. This is a replication bubble. When replication bubble is formed, then RNA polymerase start its work. It join the nucleotides, okay, opposite sequence to the opposite sequence of the template strand. Sorry, I not tell you about these two strands. This is 5 prime and 2 3 prime end strand, and this is 3 prime and 2 5 prime end strand of the DNA. Okay, where on which strand RNA will be synthesized, on which strand RNA will be synthesized is called template strand, and the opposite strand of the template strand is known as non-template strand or coding strand or sensory strand. Why it is called coding strand? Because the sequence of the nucleotide present on the non-template strand is approximately same to the sequence of nucleotides on the newly formed RNA. Okay, but there is big difference between the non-template strand sequence of the non-template strand and the sequence of the RNA. Non-template strand contain thymine, but RNA contain uracil in the place of thymine. This is the difference between them. Okay, it starts when RNA polymerase attached to the promoter site. It break the hydrogen bond between the nitrogenous bases and replication bubble is formed. Now it starts its work. These are the sequence of nucleotides or nitrogenous bases on the template strand. RNA polymerase will join the opposite nitrogenous bases on this sequence of the template strand, like this, and will join it. Now, a small portion of RNA has synthesized. Okay, when small portion of RNA has synthesized, it is about 9 to 10 nucleotide long. then sigma factor release the RNA polymerase because its function just was recognition of initiation or recognition of starting point of transcription. Okay, now it released the RNA polymerase and remaining portion of RNA polymerase will called core enzyme. Now core enzyme is present on the promoter side. Now a structural change has occurred in the RNA polymerase. Due to this structural change, RNA polymerase become coenzyme and lose its weight. Due to the loss of weight, it is capable to move now. Now it is capable to move because the loss of weight. Okay. Now it starts moving to the 5' end of the template strand. Okay. When it moves from promoter side to the gene side, it will look like this. Now RNA core enzyme has reached at the gene region. Okay. Now it started its work. This is the promoter side, this is the terminator side, and this is the gene. Now it started its work. This is the piece of RNA which is synthesized in the initiation. Now elongation has going to occur. Okay. Initiation has occurred. Initiation is finished. Now elongation is start. Okay, this portion was synthesized in the initiation step and it is hanging. Why it is hanging? We know that RNA has uracil in the place of thiamine, and we know that adenine present on the template strand form weaker bond with the uracil, okay, as compared to the thiamine which is present on the non-template strand. So adenine thiamine bond is dominant on adenine uracil bond. That's why bond between the uracil and adenine will break and rejoin adenine with the thiamine of the non-template strand. Okay, that's why RNA piece will start hanging. Okay, now there is a sequence of nitrogenous bases on the gene portion. And coenzyme started its works. It joined the opposite nucleotide, opposite nitrogenous bases here. Now elongation has occurred because a long piece of RNA has synthesized. Okay, and when this portion will be copied, coenzyme starts moving. Okay, it is now capable, also now capable to move forward. When it moves from gene region to terminator region, it will look like this. Now I am raising this diagrams. Now elongation has occurred, initiation has occurred. Okay, now we are going to termination, the third step of the transcription, termination. There are two types of termination basically: row independent termination, row independent termination, and row dependent termination. Row dependent termination. First, I am going to discuss about the row independent termination. Okay. Now, coenzyme has reached at the terminator site. This is a gene site which has copied. Okay. After this, it is a promoter site. Now, coenzyme has reached to the terminator site. Gives specific sequence of nucleotides. This sequence is complementary to each other. What is meant by invert complementary to each other? Look at this. 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4. 1, 2, 3, 4, 5, and 6. Okay. If there is guanine, and there is cytosine, thiamine, adenine, thiamine, and guanine. Here will be invert complementary to this sequence. If there is guanine, here will be cytosine. If there is thiamine, here will be adenine. If there is adenine, here will be thiamine. If there is thiamine, here will be adenine. If there is cytosine, here will be guanine. If there is guanine, here will be cytosine. Okay. Here will be n type of sequence like X, suppose X, X, X. Okay. Now, coenzyme start its work and place the opposite basis, nitrogenous basis, to this sequence of DNA template. Sorry, I write the letter here. If there is cytosine, what will be here? Guanine. If there is guanine, here will be cytosine. If there is adenine, here will be uracil because thiamine is absent in the RNA. That's why here will be uracil. If there is thiamine, here will be adenine. If there is adenine, here will be uracil. If there is cytosine, here will be guanine. Okay. If there is X, here will be Y. If there is guanine, here will be cytosine. If there is thiamine, here will be adenine. If there is adenine, here will be uracil because we know that thiamine is absent in RNA. If there is thiamine, here will be adenine. Here, if there is cytosine, here will be guanine. If there is guanine, here will be cytosine. Okay. This is the portion of RNA which is synthesized in the gene region, and this is the portion of RNA which is synthesized in the terminate region. Okay. Due to this invert complementary sequence of nucleotides or nitrogenous bases, it will it will pull down, and hairpin loop-like structure will form like this. Like this. Because of invert complementary sequence, it will form hairpin loop-like structure like this. Okay, this sequence of 6 nucleotides will be here, and this sequence of 6 nucleotide will be here, and this sequence of 4 nucleotide will be here, like this. 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4. Okay. What will be sequence here? Cytosine, cytosine, adenine, adenine, uracil, uracil, adenine, adenine, guanine, guanine, cytosine, cytosine. Okay. If there is cytosine, here will be guanine because this is guanine. Okay. Guanine, again guanine, uracil, uracil, after uracil, adenine, again uracil, cytosine, and guanine. The sequence of these six nucleotide is complementary to each other. Okay, that's why hydrogen bonding will occur here between them. Here will be Y, 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 and Y. Okay. Now, after this sequence, after this invert complementary sequence, a continuous arrangement of adenine comes. Adenine basis comes. Adenine, 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 and so on. It may 10 to 20 or 30. Okay. If there is adenine, continuous arrangement of adenine, then RNA coenzyme will place uracil to the opposite side. Okay. Uracil, 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 like this. Okay, and we know that the hydrogen bond between the uracil and adenine are very weak. Okay, and this process is very occur in very fast speed. Okay, and elongation has occurred. Due to the elongation, the weight of the RNA strand has increased. Okay, due to the weight and the speed of the reaction, a sudden jerk is produced. A sudden jerk is produced. Due to this sudden jerk, the weak hydrogen bond between the uracil and adenine will be broken. Okay, will break, and the part of RNA will separate from the DNA template. In this way, row independent termination occur because there is no need of any type of protein, any assistance for the breakage of the hydrogen bond between the uracil and adenine, and for the separation of the RNA from the DNA. Okay. Now this is the type of termination and row independent termination. What will be the definition of row independent termination? The termination in which separation of RNA from the DNA template DNA strand occur without any row protein is called row independent termination, or we can say that the termination in which row protein is not needed is called row independent termination. Now I am going to tell you about the row dependent termination. In row dependent termination, same procedure will occur, but there will be no continuous arrangement of adenine. Here will an other type of arrangement of nitrogenous bases. It may be guanine, cytosine, adenine, thiamine, guanine, cytosine, adenine, thiamine, etc. Okay, and coenzyme will place opposite nucleotide or opposite bases according to this sequence. If there is guanine, here will be cytosine. If there is thiamine, here will be adenine. If there is adenine, here will be uracil. If there is cytosine, here will be guanine. If there is guanine, here will be cytosine. Now, the hydrogen bonding between these nitrogenous bases are very strong. Now, due to the speed and weight of the RNA strand, a sudden jerk is also produced, but this sudden jerk is not capable, is not enough to break these strong bonds. Now, we need something to break these bonds. That's why a row protein attached to the RNA strand. This is row protein. Row protein can move, and coenzyme also can move. The speed of movement of coenzyme and row protein is approximately same. It starts moving. When it reaches to the hairpin loop-like structure, it also break the hydrogen bond between these nitrogenous bases because it also act as helicase enzyme. And we also know that what is the function of helicase enzyme? It break the hydrogen bond between the nitrogenous bases, and it now row protein is performing its function. It also breaks the hydrogen bond between the nitrogenous bases of the hairpin loop-like structure, and when it reaches to this point, it also breaks these hydrogen bond between the nitrogenous bases, and RNA strand will separate from the DNA template strand. Okay, and termination occur with the help of row protein. That's why it is known as row dependent termination. So, what will be the definition of row dependent termination? The termination in which row protein is needed is called row dependent termination. This was the lecture. I hope you all understand, and I try my best to understand you. I request you to subscribe my channel and like my video and share it with your friends. Assalamu alaikum.